I'd like to welcome all the members of PARC and guests. Also like to extend a special welcome to the PARC graduate students and postdocs and any other people online who are joining us that way. Today we're pleased to have Professor Sergei Savikhin, who's an associate professor of physics at Purdue University, joining us. Sergei is a native of Estonia and got his degrees, all his degrees at the Institute of Physics, I guess it's Tartu University in physics, working with Arvi Freiberg, who may be known to some of the people in the room. A person who's worked in photosynthetic antenna complexes for many, many years, and a good friend of many of us. So after Sergei finished his degree with Arvi, he moved to Iowa State University Ames Lab in 1992 and worked with Walter Struve. And that's when I came to know Sergei, and we have several papers from that period together, which were quite interesting. And then in 2001, he moved to Purdue, where he has been ever since, and has done some very nice work at Purdue. His research interests include femtosecond optical studies of artificial and natural biological systems and various biomimetic devices. And also, he's really an instrumental genius in terms of setting up new types of ultra-fast laser experiments. So, Sergei, thank you for coming to visit.
Does it work? Okay, good. Thank you very much for very nice words, especially the genius part. Very surprising. And yes, we had some good time with working with Bob in the past, and hopefully we are now collaborating again, and we'll get some good results out of that work as well. Now, what I would like to talk today about is not exactly about energy transfer or energy conversion processes in these photosynthetic proteins, but I would like to talk about unconventional photoprotection mechanisms in photosynthetic proteins. So this is something which we often forget, but these systems are really working very hard to survive in the sunlight and in the oxygen we have in the atmosphere. Now, what I would, can we go down with lights a little? Do you see that well? Or kind of not very. Anyway, so this is outline, so I'll give a very short introduction. Thank you. And there will be several parts in my talk. I will talk about photoprotection in cytochrome B6F protein, which is photosynthetic protein. In particular, we discovered two new photoprotection mechanisms in this cytochrome B6F protein. One is electron transfer mediated chlorophyll A excited state quenching, which is singlet excited state quenching, which protects it from being killed by light and oxygen. And the other one was a very surprising finding that there is just a long distance triplet energy transfer possibility in the protein, which can also photoprotect chlorophyll. We will also talk about Chlorosome, which I worked before with Bob on electron or energy transfer in this complex. But we were up to 200,000 pigments in it, and the photoprotection should be extremely important. And we found that there is kind of unconventional, I would say, method to protect this system, and it involves triplet exciton formation. And on a side, I will, at the very end, I'll talk about oxygen in general and oxygen content in a functionally photosynthetic cell. So how much oxygen really gets into the cell because it is produced there. And that I will discuss when this system had to develop the photoprotection method, because as I will show you, in the very beginning of evolution, when there was no oxygen, basically everything was pretty stable. So this is obviously known reaction to everyone in this community. So this is what photosynthesis is about. Take light and run this simple chemical reaction, and basically store the energy of light in energy of chemical bond. Now, this shows this is basically plants photosystem, or some bacteria has the same, shows several major protein systems which are involved in photosynthesis. It's photosystem 2, which all it does is basically split water in response to light, so it uses energy of light absorbed by the system to produce protons to oxidize water, and also oxygen is vented into the atmosphere. Oxygen is not used by that system. Now, the protons which are produced during that will be accumulated on one side of the membrane. They can't penetrate through the membrane, so basically, I'm a physicist, for me, it looks like charging a capacitor, so we are getting these electrical energy. The electron which is removed, or electrons which are removed from oxygen, go to the other side of the membrane where they are picked up by phosphochinone molecules. It picks, each one of those picks 2 electrons and also picks 2 protons from the other side of the membrane, and transport that 2 protons to the other side of the membrane in cytochrome B6 complex, which we will be talking about briefly. So here it releases the electrons and releases protons, again, adding to the proton pool on this side of the membrane, on the inside or luminal side of the membrane. It also couples these electron transfer fluids to the proton translocation across the membrane, and I will cover that in the next slide. So the electron goes all the way through the phosphocyanine, which picks it up from cytochrome B6, brings it to photosystem 1 complex, which again acts in the same way as photosystem 2 in a sense. It absorbs light and uses that electronic excitation energy to boost the energy of electrons, transport it to the other side of the membrane where it is picked up by pyridoxine, small protein, which brings it to FNR, to an ADP-H reductase, which basically runs this chemical reaction, making an ADP-H molecule out of an ADP plus and protons. And of course, the protons which were accumulated during this process on one side of the membrane, so basically the charge of this capacitor is driving ADP synthase, which is basically molecular size mechanical motor, which converts that electrical current, motion of protons, into motion, and uses that mechanical energy now to grab ADP and CI group, and basically use mechanical energy to push these 2 molecules together, making ADP the universal energy carrier. Now, the working force of photosystems 1 and 2, and actually of many other antenna systems, which I didn't cover here, is of course chlorophyll or chlorophyll-like molecules, and chlorophyll and so on. Now, it is chosen by nature long before there was oxygen in the atmosphere. And it was chosen for a very good reason. It is a very good absorbent of light. And it has some other electronic properties which make it perfect for this use. So it can absorb basically all light between 600 to 700 nanometers, and in blue region, if you add a few carotenoids or some other molecules, you can also take care of this part of the spectrum. Now, its oscillator strength is extremely high, so it's about the order of 5 or more dpi, which is a very, very good absorbance. And when nature was picking that molecule for photosynthesis, obviously that was the reason why it picked it. And if I would say today to an engineer, would you pick a chlorophyll molecule to make something which is light sensitive or accumulates light, engineer would laugh at me because it turns out that this molecule is extremely unstable. Now, why is it unstable? So if you have a chlorophyll, there are 3, actually not 3, more states, but the major transition is transition from S dot state to S1 state. The electron goes to excited state, it's called also QY state. That's about 700 nanometer photon, or 670 nanometer photon for chlorophyll A. So it ends up in S1 state. Now, the energy is stored here for about 5 nanoseconds if you just have the chlorophyll molecule. After that time, it can either return back and emit a photon, or it can form a triplet state. Now, in triplet state, the spin of electron flips, and now the transition down to the lower level is forbidden. So this state lives pretty long time. So the lifetime of that state is in the order of a millisecond. And Blanket just published a paper like a year ago measuring all 10 known chlorophyll. So that was like in the order of 400 microseconds, I believe. Now, this is all very nice, but the problem is that the energy of that state is such that if oxygen molecule happens to be nearby, it just bumps into it, the energy could be transferred to the oxygen, creating singlet oxygen, which is an excited state of oxygen. Singlet oxygen is very strong absorbent, or very strong chemical agent, so it can actually oxidize everything around much better than oxygen itself, because its lower energy level here is now empty, so it can accept electrons from anywhere. So it's a much stronger oxidant. Now this time is basically defined by the diffusion of oxygen. So, and in normal condition in water, it will take about 1 microsecond before an oxygen, in a saturated with atmospheric oxygen water, can bump into the chlorophyll, gets close enough within basically touching distance, and the energy transfer is extremely efficient. So instead of going back smoothly and emitting or going into heat, this energy goes into producing singlet oxygen. Singlet oxygen can destroy the chlorophyll itself, or oxidize that, or it can oxidize any protein around, so basically anything organic around. The lifetime of chlorophyll in solution under sunlight in these conditions would be in the order of 5 minutes. Just a few minutes and it's dead. Obviously, no one in right mind will use that molecule to absorb light and basically serve as a vehicle for energy conversion, light energy conversion. Now, to deal with that, the conventional understanding or a conventional mechanism proposed and tested many times is that there are carotene molecules very close to chlorophyll, and the energy of a triplet state of carotene is somewhat below that of the chlorophyll triplet state energy and below that of the oxygen triplet state. And that's important. If it's close enough, it will take about 1 nanosecond time for energy to be transferred, triplet energy to be transferred from chlorophyll to carotene. Once it's on carotene, it can create any of the singlet oxygen, so it's pretty safe, and it takes relatively short, like 3 or 10 microseconds to basically bring it all down and transform that energy into heat, which is relatively safe. Now, not all systems actually use the exact same mechanism, and one of the exceptions which we found several years ago was cytochrome BChl. Now, this is the protein for which structure was actually determined at Purdue University by Bill Kramer in 2003, and that's probably why I became interested in that protein. Now, the function of that protein doesn't really require the presence of chlorophyll. So what it really does is simply accepts plastoquinol. So somewhere here is a docking site for plastoquinol. So, and the, actually, this is inverted, sorry. So plastoquinol comes into these docking sites, and it releases 2 electrons. One goes up to the P or N side of the membrane, picked up by these schemes. And here another plastoquinol comes in, and it basically uses that electron to capture 1 more proton. Now, the second electron here goes into the cytochrome F complex, which basically this is the stripped version of the cytochrome B6F without the protein. So it goes to here, and then it's picked up by plastocyne. So this way, the system basically doesn't do anything else but just transfer of electrons and replicating proteins. There is no need for any kind of a chlorophyll. Light is not involved in its function in no way. However, once the biochemical analysis were done, it was found that there is a chlorophyll molecule there inside of this system. Now, the function of this chlorophyll molecule is still debated. So this is where it is in the structure. So that's basically its pocket which it occupies in the protein. So it has been proposed that maybe it does light harvesting long ago, but obviously it doesn't. This protein is too far from anywhere else, so this chlorophyll simply does not communicate with another antenna system. So just suppose maybe some antenna system is closed and you can transfer it. That doesn't happen. So electron transfer, it doesn't involve, no experiments show any involvement of chlorophyll in electron transfer, which is the main function of the cytochrome B6F. There are a bunch of other functions proposed, which are just listed here. But that's not so important for the talk today. What I was interested more in is that if there is a chlorophyll A, this is a potential way to damage the system, because we know it can damage the proton. Now, if you have a chlorophyll there, for whatever reason, you have to protect the system from possible damage. And indeed, biochemical analysis showed that there is beta-carotene in the system. And it was, of course, automatically proposed that this beta carotene has to be very close to chlorophyll in order to protect it. It really has to touch it in order for triplet energy to be transferred efficiently from the chlorophyll A to beta carotene. However, when structure came out, it was a complete surprise because the distance between chlorophyll A and beta carotene, this is just a zoom in picture, that was 14 angstroms. Now, 14 angstroms, if you do antenna systems and energy transfer, that seems to be pretty close. So the dipole-dipole interaction with first order energy transfer occurs very fast. However, triplet energy transfer doesn't follow the same rules. As a matter of fact, there is no basically dipole-dipole interaction involved. And what you have to use is a second smaller, usually term, which is usually smaller, so this is a Dexter type of exchange mechanism, which involves basically electron tunneling through empty space, or through the space between the molecules. So what happens, instead of in first order kind of energy transfer, what happens, the donor returns to ground state, so electrons from upper excited state falls out down, and at the same time, in acceptor, the electron goes to excited state. There is no electron exchange. In this system, in this kind of mechanism, however, this is impossible because that condition is not allowed. So what happens instead is that these electrons basically tunnel through the space like this. So one electron goes to the acceptor from the excited state, and the ground state electron simply returns to the donor. So this is a very cartoonish representation of these mechanisms that occurs there. Therefore, it's governed by electron transfer rule, which is exponential dependence on distance. It's basically overlap between the wave function of the electrons on two systems. So this is exponential here. So this is the rate of energy transfer, triplet energy transfer, and it depends exponentially on the distance between the molecules. And 2 comes from the fact that we have to transfer 2 electrons at the same time. So, very nice. If you do calculations, you'll find that indeed, if it's a 4 angstroms between the molecules, between chlorophyll and carotenoid, the transfer time would be in the order of 10 nanoseconds, shown here. Now, this is 14 angstroms. And if you do the calculations, what you get is that the transfer should occur within about 300 microseconds. Now, if we have oxygen in the system, that will take it in the order of 1 microsecond. Obviously, there is no competition here. I mean, there is a clear win for production of singlet oxygen instead of basically quenching it. Now, this was the state at which I got it, because I moved into that, I wanted actually to see if there is a triplet energy transfer. And while I was setting up my experiment, the structure came out, and it was basically useless to do the experiment. But still, since I was already on that track and prepared to do experiments, we did the experiment. So we tried to excite chlorophyll A with our laser, and we wanted to test if the triplet state which will create here can actually reach carotenoid, expecting the negative answer, obviously, to that. Now, this, I skipped. So this is actually the light-induced irreversible degradation of chlorophyll, just to show you how much more stable the cytochrome B6F than the chlorophyll A in solution. So this is the chlorophyll A in ethanol that takes about 6 minutes or 2 lifetimes, but in the order of 6 minutes to degrade it, as I already told before. And if you use chlorophyll A in B6F, it will take about 14 hours to photodegrade it under the same conditions. So obviously some kind of photoprotection exists, even though that the carotenoid is so far away. Surprisingly, in spite of the fact that carotenoid is so far, we could see the carotenoid triplet state. Now, besides that, before we did that experiment, we also considered another phenomenon, which is that the triplet state yield would really depend on the lifetime of the singlet excited state. Now, in the antenna systems, there is a rapid energy transfer from one chlorophyll to another one. So the excited state doesn't leave for 5 nanoseconds. And since it doesn't leave for 5 nanoseconds, there is a little chance that it can form the triplet state. So it's pretty much proportional to the, or inverse proportional to the lifetime. If you can move away the excitation from the singlet state, then you obviously make much less of a triplet state and much less of a singlet oxygen. So we had to measure the lifetime of that chlorophyll. Now, one expects, since it's just one chlorophyll, there is no energy transfer from that chlorophyll to anywhere else, that lifetime of that chlorophyll should be in the order of 5 nanoseconds, as in the solution. So we set up to do that experiment. So this is basically a pump probe experiment, something that many of you are familiar with. Briefly, we use a pump pulse to create this one state. And then we use a probe pulse to measure the absorption from that state. If some of the molecules are in excited state, it would be a triplet state, whatever, they are not in the ground state, they will not absorb. Therefore, we'll see the bleaching of the absorption. And we can probe that with another pulse. So this is done in our laser lab, which is basically femtosecond laser lab, unlike your lab here, this is all home built. So it has a bunch of laser stands and a bunch of mirrors on the table, but it works fine, producing about 100 femtosecond pulses, which is good enough for what you want to do. And besides, it has a very good delta A sensitivity. The absorption difference can go down to 10 to the minus 5, which means that even if we change the state of just one molecule out of 100,000, we can basically start seeing the changes associated with these molecules. So this is the result of the experiment. Surprisingly, instead of 5 nanosecond lifetime, we saw something like a 200 picosecond lifetime. So this is the initial bleach of absorption due to the fact that we move chlorophyll into excited state. Then it drops to triplet state, and actually it doesn't. It comes back mostly, and that's what we see here. The triplet state yield is relatively small because it spends only about 200 picoseconds in that state. Since it's only 200 picoseconds, not 5 nanoseconds, we will reduce the fraction of the triplet state which is created by every single quarter in the system. So this is basically the difference between 5 nanoseconds and 200 picoseconds. So it's about 25 times shorter. So there will be triplet yield, which is about 25 times smaller than in chlorophyll, which is just in the solution because of that. Now, there is no energy transfer from that system. So the question is, what is really quenching the singlet state of the chlorophyll? Typically, it's just energy transfer or electron transfer. Now, surprisingly, we did all kinds of experiments and research on that, and what we found is that the mechanism for the quenching of this chlorophyll in the system is really electron transfer. Even though it doesn't participate in electron transfer, the mechanism for quenching is connected to that phenomenon. So to illustrate how it happens, I have drawn here this very simplistic picture of a chlorophyll and, let's say, nearby residue, pyrazine, any one of those. Actually, it's not pyrazine, as we figured out later, but the mechanism is the same. So this is a home of highest occupied molecular orbital. So it's electrons in the ground state, the highest one. This is in pyrazine, this is in chlorophyll. Now, this is the LUMO, which means lowest unoccupied molecular orbital. So basically, whenever there is a simple transition, when chlorophyll absorbs light, the electron goes simply to excited state, leaving coal behind. And this is what will happen. So at time 0, we will shine light on it, and that will basically bring electron up to the excited state. Chlorophyll becomes excited. That's very simple. Now, in the redox potential diagram, it basically means that the chlorophyll moves to the lower, higher end of that, plus 1.2 volts or whatever it is. So these are actual real volts which are measured by someone else, showing that it is actually visible, the mechanism. Now, once the electron is here, what can happen next is that instead of these electrons coming back, we can have electron transfer from nearby residue, like pyrazine, tryptophan, and phenylalanine are actually good candidates for that due to energetics. And this electron will simply transfer physically, tunnel to the chlorophyll, making chlorophyll minus and pyrazine plus. Now, we did some calculations for that system, and indeed, you get in the order of 200 picoseconds very nicely for this particular process. Once it moved here, of course, the chlorophyll minus will produce also a signal, but it turns out that the lifetime of chlorophyll A minus here is very small because these electrons now tunnel back into that state here, bringing it to basically ground state. So this kind of a quenching mechanism had been actually detected or found earlier to exist in riboflavin containing proteins. So that was already published, so that's how we came to that idea at all. So this is, again, the calculations show that it matches perfectly. We tried to do some mutations to figure out which one of these residues tyrosines first because energetically it seems that they are most visible. Now these mutant replacements didn't actually work, so nothing changed. There is still 200 picosecond lifetime. We also tried that tryptophan, and that didn't work either. We tried some other mutations, but those mutations simply didn't grow, as it often happens, so something really goes wrong. So actually, by calculations, it's probably this phenylalanine 102, which is 5.5 angstroms away, because one has to take into account all the electric fields which exist in the protein itself, do all the residues, and it seems that there is a basically vector force which would drive electrons in this direction between these two molecules. Now, this is all nice. We got 25-time protection due to that quenching method, but it's still not the observed 130, 140 times, which we see compared to the chlorophyll. So there should be some missing 5 to 6 time photoprotection mechanism in that system. And that's where we actually did this experiment for the energy transfer, even though we didn't expect any energy transfer, but just to show that there is no triple energy transfer between chlorophyll A and carotenoids, which are 14 angstroms apart. So we pumped into chlorophyll A, really pumped a lot, so we know that we are creating some at least, and then go to triplet state. And then we looked at the area at about 520 nanometers for the signal changes, and it is known that at 520 nanometers, if you create triplet carotenoid, it will have extra absorption there. And the lifetime of that triplet state is about 3 microseconds. Surprisingly, this was exactly what we saw. This is the additional absorption at the wavelength, which represents basically the triplet state of the carotenoid, and it indeed decays within about 3 microseconds. So the signature was absolutely clear. So this also didn't have any rise time here. So you would expect if lifetime is longer, then our resolution was about 10 nanoseconds. We couldn't see any rise time. So the triplet state of beta-carotene was created within less than 10 nanoseconds after we created the triplet state of chlorophyll A. So it's extremely fast. This is something which could happen if we have carotenoids within about 4 angstroms away from chlorophyll A, but not so far. There's no way to do that because we did some calculations. That should be 300 microseconds. There's no way we could see that. That was a surprise. We, of course, tested also the 670 nanometers, where it should be a signal from the triplet state of chlorophyll. We didn't see any, so it was just a flat line. Everything, all the signal was at 520 nanometers immediately. So we couldn't create any triplet state of chlorophyll. We did create it, but we couldn't observe it because it just transferred the energy so fast to the carotenoid. And this actually is the K-associated spectrum for this about 3 microsecond component, which is associated with carotenoid, and it showed a very nice band at 520 nanometers, exactly as expected for carotenoid. There is no other explanation. It is really created. So the question was, how can we transfer triplet energy over such a long distance in such a short time? So we can't do it directly. There is no way electrons can actually tunnel through that space. 14 angstroms. If you calculate it, it never works. So the hypothesis was that the third molecule, X, mediates energy transfer from chlorophyll to beta-carotene. So we need a third molecule. Now the third molecule could be also mobile. It could be actually moving, because otherwise we will actually need more than one molecule in between. So it should be several, a chain of molecules to transfer to the state. So it's easier to assume that there's some mobile molecule. And the best candidate for that, so this would be how it transfer goes to X excitation, and then X transfers to carotenoid, making triplet state of carotenoid. So best candidate for X is actually oxygen itself, because oxygen readily takes triplet energy from chlorophyll. Now, of course, kind of not the safest way to do it, because the oxygen species are pretty reactive, especially if you make this X star molecule, which will be now oxygen singlet state of the singlet oxygen. Now, but this is the diagram, and I will show supporting data later. So this is a conventional scheme for photoprotection. You put a carotene really very close to the chlorophyll, and you just quench it very fast. Now, what we proposed is that we swap these two. So we first go to oxygen, nicely, create singlet oxygen, and then singlet oxygen is diffused by carotene. As a matter of fact, this phenomena is known to exist. If you will have singlet oxygen production somewhere in the solution. So you put some synthesizer for that, and you put carotenoids in. The carotenoids will actually readily quench the singlet state of oxygen. If oxygen diffuses, bumps into carotene, it just releases its energy and it's diffused, basically. And that happens relatively fast. Now, this is then what should be happening. Light comes in, we create excited state, triplet state. Now, oxygen picks up the triplet excitation, travels inside of the protein, and diffuses inside the beta carotene site. And it goes back, and the process repeats and repeats and repeats. Now, the argument against that would be, of course, on the way of doing so, diffusing to that, it should be probably oxidizing everything else around. Luckily, we found nice chemical information about the chemical reactivity of singlet oxygen towards all the proteins which are inside that system. And it turns out that reactivities of singlet oxygen toward these amino acid residues, which are on these pathways, is about 100 to 1,000 times lower than reactivity towards the beta carotene. There's a much higher chance to be quenched by beta carotene than actually do some damage to the residue. So that's known. So it is possible that this really happens. Now, second evidence was the structural evidence. So we looked at the structure. This is the basically cross-section between the carotenoid and chlorophyll, and these blue ones are actually hydrophobic residues. Now, the hydrophobic channel really, the oxygen is hydrophobic, so it really loves to go into hydrophobic channels. And such kind of channels are, for example, a C oxidase protein is known. It has about 14 angstrom channels for oxygen. And there were some modeling done for these oxygen diffusing within that channel, which is 14 angstrom long. And within 2 nanoseconds, the oxygen in these modeling actually diffused many, many times through the length of the whole channel. So it was extremely, extremely fast. It was a kind of interesting piece of data, which also helped us to identify that mechanism in this particular protein. Now, the next one, if this is oxygen, it has to be mobile. So the one thing is, these are experiments we want to repeat. And these are the first experiments which we did some time ago, and we didn't repeat them. But this is low oxygen mobility, which could be achieved at low temperature. So we freeze the sample, the oxygen should move so fast. Now, this is room temperature, so this is the 3 microsecond lifetime of the carotenoid, and this is the 520 nanometer wavelength. So it actually quenches very fast, as expected. As we go, let's go to 10 Kelvin immediately. So at 10 Kelvin, we don't see anything which is very short. There is a little bit of a short component in front, which could be still some kind of a triplet carotenoid form that we need to confirm. But the major thing is that we see now long signature, which is additional curves simply from the triplet chlorophyll. So the triplet chlorophyll just stays now. So as it's a cold temperature, a lot of that triplet chlorophyll doesn't disappear anymore. So it doesn't quench. There is no oxygen mobility, so the triplet state of chlorophyll stays. Low oxygen concentration is something which is very hard to do, especially because that pocket there is hydrophobic and oxygen likes to stay there. So what we tried to do was an experiment which involved xenon, and xenon is known to go into places where oxygen goes. So we basically got oxygen out and we tried to put a high pressure xenon in, and it was supposed to replace oxygen in those spots. In this case, this is the aerobic 3 microsecond lifetime of the carotenoid. And then we did the xenon treatment, which wasn't very easy here. It's a very noisy curve. But there is certainly nothing in the order of 3 microseconds anymore. There is something much longer and much lower intensity. Again, it shows clear dependence that the oxygen, if you remove oxygen, this lifetimes change, and we don't create or keep much of the triplet state of carotenoid. So, again, these are experiments which we are currently reproducing to get them cleaner. Now we know how to get more oxygen out from the system, which is typically hard to deoxygenate samples. Now, the other part of evidence is that these xenon-treated samples, the X-ray structures were taken by Bill Kramer, and what occurred is that indeed the xenon shows very nicely an X-ray structure, and they show that it's located right next to the chlorophyll. So it was indeed within the reach of the chlorophyll, so it could So this was basically twofold, two different mechanisms to photoprotect the chlorophyll inside of that protein, which haven't been reported before. As a matter of fact, these long-distance triplet energy transfer may be working in other systems. For example, in photosystem 2, the carotenoid is about 14 angstrom away from this special pair. Now, seems to be no photoprotection, but as we do a Foldsworth measure of the lifetime or the transfer time between those within about 100 nanoseconds or less. This needs to be checked, but it was much faster than possible for direct transfer of triplet space. So this is another example where it could actually work. And of course, there might be many more proteins where the long distance transfer, triplet energy transfer is working. The function of that chlorophyll is still unknown. So I can only speculate about that, but this is not the topic of today's talk. Now, this is one system I wanted to talk about, and the other system is something which I have loved for a long time, from the time we worked in the chlorosome. So the chlorosome itself. The chlorosome has a 200,000, up to 200,000 bacterial chlorophylls in it. And they're closely packed, and obviously it should be very sensitive to oxygen and light, so it should photoprotect itself. Now, since it has so many chlorophylls, it's of course very efficient in light gathering, and basically can survive under a condition where one photon is absorbed by a single chlorophyll molecule in 8 hours. So chlorophyll gets 3 photons a day, and that basically makes it stay very happy. They grow. Of course, these bacteria divide in what? Multiplying like 3 years, so even more. But anyway, so these are quite amazing complexes, and this is something which you found, I believe, that they actually grow at the hydrothermal vent and the depth of 2.44 kilometers where they're in the water, where there's essentially no light at all from sun, but what it uses apparently is the black body radiation of the vent itself, which is quite impressive. Anyway, so this is the schematic structure of that, taken from Sugar Don Bryant's webpage, as many people do, take this picture. So these are the bacterial chlorophyll T rods, which are within the membrane. There's also the base plate with the PSMA protein, which basically transfers energy from the bacterial chlorophyll C system to the FMO, and then it goes to the reaction center. Our interest was, of course, that big blob here, which contains a lot of bacterial chlorophyll Cs. It also contains about 10% of the concentrated carotenoid. It also has quinones, about 10%, 1% of bacterial chlorophyll A, this is this base plate, and about 10 different proteins in the membrane. Now, the function of these carotenoids was, of course, suggested to be photoprotection. And that's what we, of course, were also sure about. However, we got access to the carotenoid-free samples grown by Don Bryant some time ago. And of course, we looked at their photostability. The bacteria themselves didn't really grow well in this condition, so you had to really have a low oxygen concentration and so on. You couldn't really get access to oxygen. But in this case, this is the wild type, so this is basically photodegradation. We took a laser, a really powerful laser, and we shined on the system, on the chlorosome, and we look at photodegradation. Basically, it's an absorption, how the absorption band goes down as we degrade bacterial chlorophyll C. Within this power, it takes about 100 minutes to kill the wild type, half of wild type species. Then we switch to carotenoid-free sample. This is PRCD. This is this curve here, and we found that it's only 3 times less stable. So it still lives for about something like 20, 30 minutes under this very strong laser excitation, just to show you what will happen to bacterial chlorophyll C, which is unprotected in solution. At this laser power we use, it will take a few seconds to kill it. This is still extremely stable, even though there are no carotenoids whatsoever. Before, there was at least some use of carotenoids, quenching the Singlet oxygen which was created. Here, obviously, we didn't need any carotenoids, and it was still reasonably stable. I'm not saying it didn't photoprotect at all, but obviously that was not the major mechanism to photoprotect the system. So carotenoids provide only limited or 3 times protection. And obviously, the major photoprotection comes from some other mechanism. So then we looked at the bacteriochlorophyll C triplet state kinetics, obviously to see if it is quenched by something else, maybe. And that shows very nicely, very long lifetime. This is like 10, 20, 30, 40 microseconds, goes long. Now, this is a carotenoid-free mutant, so there are no carotenoids, nothing goes to carotenoids. And obviously, then triplet state, if it's not quenched by carotenoids, by anything else, it just stays for many, many microseconds, at least in the order of a millisecond. Moreover, there was no oxygen dependence. The black curve is taken without oxygen. The blue curve is taken with oxygen. So the oxygen obviously didn't influence this triplet state at all. So there was no energy transfer to oxygen, which was very, very strange. Then we went to the wild type, which is shown here. It's a different scale, but it's the same lifetime. And it looked exactly the same, with oxygen, without oxygen, and even with carotenoids. So there was no transfer of triplet state from this triplet state of bacteriochlorophyll C to carotenoids nor to the oxygen molecule itself. And it was stable. So this is how it skipped. So what are the possible mechanisms for this kind of a triplet state which doesn't transfer energy anywhere? The first thing we thought about was, of course, there are kinones. And maybe kinones do some kind of electron transfer back and forth and somehow photoprotect the system. Now, that turned out to be dead end because we changed the redox state of the kinone and nothing changed at all. Everything stayed exactly the same. And then we thought that these pigments are very close to each other. And it's known that excitonic interactions in such systems are very important. So, for example, if you look at the singlet-singlet interaction, so this is a typical calculation of excitonic state. So this is the bacteriochlorophyll C absorption, which is maximized at about 670 nanometers. Once you assemble those into those aggregates, you get it shifted by about 80 nanometers. So this is the chlorosome absorption section. So, and this reason for that shift is, of course, the excitonic interaction. They're very close to each other. The distance between the two pigments in that system is about 3.5 angstroms. So the interaction, dipole-dipole interaction, is extremely strong. And this is why we have these enormous red shifts. This shows underlying quantum mechanics for calculating this state. So the separation between the states basically depends on the energy of original state, that's the separation, and the interaction energy between the pigments. In the case of two pigments, but in the case of many pigments, like here, 200,000 pigments, so this Hamiltonian energy system will have 200,000 by 200,000 thirds in general. So we thought, okay, that's a very plausible explanation, because if we bring the energy of a triplet state down, as it happens here, but for triplet state, if it goes below that of oxygen, then there will be no energy transfer, because it's just energetically not feasible. So this is the actually model with all the energies. This is the energy of a triplet state of bacteriochlorophyll C, 1.29 electron volts. It was measured before by someone else. So this is the singlet oxygen energy. So the 0.97, obviously, it goes very nice. And this is the carotenoid energy, which is below that of oxygen, as it should be for photoprotection. Now, if we have interaction between many triplet states, many bacteriochlorophyll states, there will be a manifold of excitonic states instead of that state. And the lowest state could be below that of oxygen. And then, of course, everything rapidly transfers with that system to the lowest triplet state. It's a triplet transfer within the same system. If this is below oxygen and carotenoid, then it will explain exactly what we see in our system. Now, we did the calculation. So this is basically a matrix. We just assumed a very simple idea. So it's a sheet of chlorophylls. The separation between them is about 3.5 angstroms. So we build the Hamiltonian for that. Each diagonal energy or energy of triplet state of each of those, if it's not interacting with anything, is 1.29 electron volts, exactly this one here. And interaction energy, we took 0.1 electron volt. Now, there is a reason for that. There was a Fleming's paper almost 10 years now ago where he did some calculation of interaction between two pi-pi bonds, 3.5 angstroms away exactly. So this is the ballpark number for these interactions. This interaction, usually triplet interaction is ignored, really, because it's exponential in distance, then that's not interesting because it doesn't do really useful energy transfer as the singlet system. Because that's where it really matters. We want to get excitation from one excitonic level to another one and so on. Here, however, it turns out to be also important because when you do these calculations now in the simple mode, do the Hamiltonian calculations, you get the energy immediately 0.89 electron volts. Of course, it's a ballpark number. We don't know what the interactions exactly are. But it becomes immediately below each of those oxygens. Because I didn't change anything here, just took these numbers, whatever are from the literature, and I got it to 0.89 electron volts. If everything goes to these levels in the system, there will be basically protected. Now it can live for a long time. It doesn't matter. It will return in some time in a millisecond or so down, but that would be safe. So it's just so this. How do you prove that it really works? Because there are still quinones and there are some other proteins inside. So I told my physicists, so we don't do chemistry or anything fancy. So, but I told my students that you can actually self-assemble bacteriochlorophyll seeds into similar complexes under certain chemical conditions. So I referred to the paper where I found it, and I basically said, go and do it. So, and I was very proud about my student, who was a physicist, but he managed to basically extract the bacteriochlorophyll seeds from chlorosomes, purify them, and reassemble those into aggregates. Now, this is shown here. So this is a monomeric bacteriochlorophyll seed, which we isolated and purified from the same complexes. So it doesn't have anything else anymore. It's a very nice section. And then he used some chemical conditions to make aggregates. And these aggregates I studied long ago. All their properties, energy transfer properties and so on, look very similar to the naturally occurring chlorosome. So it's the same kind of shift and everything. So then we looked at the stability of these aggregates. They don't have anything else, just bacteriochlorophyll. And this is so this is photodegradation under strong laser light for monomeric chlorophylls, and these are aggregates. Now notice this is much faster, but it's also 2 milliwatts laser power here, and there's a 200 milliwatts here, because otherwise we just didn't have enough time to wait until we kill this system. They were extremely, extremely stable. So these are like 10 times here. It's 1,000 times more stable, photostable. So the strongly coupled systems of pigments are actually intrinsically protected by this triplet exciton formation. So this is also showing the lifetime of aggregates. So you make the triplet state in the aggregate, and it just stays there. It doesn't go anywhere. It becomes actually pretty long. And this is monomeric in methanol, so it's extremely fast. This basically transfers to oxygen. In both cases, we did have oxygen in the system. So there is no transfer of this triplet state energy to oxygen whatsoever. Now, so finishing that, I would like to spend a few moments talking about evolutionary aspects of photoprotection. Now, I got interested in that because there is always, Bill Kramer always asks me, what if it should do experiments with those membrane croppings, photosynthetic membrane croppings under elevated oxygen concentration, because in a natural system, they produce oxygen right in the cell, so there should be a lot of oxygen, more than in atmosphere. So we probably should put more in, because oxygen is an important molecule, chemically active. It might change some function or lifetime and so on. So we decided to calculate what the oxygen concentration is in a functioning cell. But on the side of that, what would be also important is that if the oxygen generated within that cell would be very high at high concentration, that raises evolutionary question. The first cyanos, which invented oxygenic photosynthesis, occurred somewhere about 2.7 billion years ago, plus minus a few hundred. So, and that's basically how the oxygen came to existence. Now, all the organisms which existed already had about, 1, 2, 3, 4, almost 500 million years, or maybe slightly less, to develop photoprotection, because the oxygen concentration in the atmosphere was going up very, very slowly. There were a lot of things which would be oxidized, so the oxygen didn't appear immediately. However, the cyanos, or the inventors of oxygenic photosynthesis, would experience this stress immediately, because that is where the oxygen is created. Now, if these levels are very high, that would mean that along with invention of photosynthesis, where you actually create oxygen, oxygenic photosynthesis, you should also provide photoprotection for all components in the cell, which could be sensitive to that. Now, this is much less probable event because you have to have a bunch of mutations at the same time. It's much easier to do one mutation, develop photosystem 2 kind of a system, and then develop the photoprotection as time goes in 100 or 200 or 300 million years. Okay, so it turns out to be very relatively simple physical task. Now, the measurement of oxygen within the cell is very, very hard. Actually, there are some attempts, but none of them are convincing because the signals are very low, the concentration difference would be low. Now, instead, we can use what we know from physics, and this is the diffusion theory. So the Adolf Fix in 1855 derived a very simple equation which shows the oxygen or flux of anything, diffusion or flux of oxygen, which is basically number of molecules which cross unit area in unit time, and that was simply proportional to the gradient in concentration and some kind of a diffusion coefficient here. That was a very simple equation. Now, we can model our cell as a spherical object. It would be similar to what we have in general for single cell organisms, and that's what we did because single cell organisms were the ones which were first created billions of years ago. Now, the flux of oxygen through that sphere could be written as shown here, 4 pi r squared, that's the area, multiplied by flux itself, gives the total number of oxygen molecules crossing that area. And of course, that should be equal to the number of oxygen molecules produced by the cell in equilibrium. Whatever is produced here should go out through that spherical area. And this basically is this equation here. You put everything together, and you get relatively simple differential equation, which pretty much any graduate or undergraduate student, or maybe even someone in high school can solve. So the answer is here. So this is the concentration of oxygen inside of the cell, given by that equation. So this is the oxygen production rate. So there's how many oxygen molecules are produced per second by the cell. And this is something which we can find in literature because this has been measured for many bacteria. And this is outside of the cell, distant r away from the cell. So it slowly goes down the concentration as the oxygen diffuses out. Now I'm showing these Globacter violosus because this is the oldest known cyanobacteria which does oxygenic photosynthesis. So this is probably the closest one to whatever was created 2 or 3 billion years ago. Now, we did the calculations using the data known from literature. These are experimental measurements of oxygen production by these cells. We use these Globacter and we use the more traditional things which everyone started, Photoprotection and Evolution. And basically, this is the size of the cell. So this is the concentration of oxygen right next to the cell or in the inside of the cell. So this is Photoprotection has the highest production rate of oxygen, most of all. And of course, the Globacter was the lowest one. It has not as advanced photosynthetic system, doesn't have as many antennas, as many reaction channels, and so on. It's very low. Now, this baseline here is concentration of oxygen in water saturated basically by atmosphere. So this is about 230 micromoles per liter. Now, this elevated concentration is basically 0.01 or 0.02 micromoles per liter above that. So it's extremely small number, really. So if you have a single cell floating in water and you shine light on it and it's producing oxygen, the oxygen diffuses so fast that this excess concentration is also only in the order of 0.01 or 0.02 micromoles per liter. It's extremely small. And I would say this is a classical theory. This has nothing to do with quantum mechanics where you calculate the absorption band, which should be at 700 nanometers, you get it at 500 nanometers, and you say, oh, this is great. I have a very good agreement with calculations. This is really precise. So you can do these calculations, and you can do the measurement. It would be simply the number plus minus 1%. So the critical concentration of oxygen for basically killing the object or killing chlorophyll will be in the order of 0.25 micromoles, because it's about 1 microsecond for the oxygen to find the chlorophyll. But the lifetime is in the order of milliseconds, the lifetime of a triplet space. So this time 1 microsecond in which the oxygen diffuses and bumps into chlorophyll depends obviously on concentration. So at atmospheric concentration, 1 microsecond. If you go down 1,000 times, it will be 1 millisecond. So it's proportional to that. And that's basically how we got these numbers. So it will be about 1,000 times less. That will be a critical number. If we have concentration of oxygen well below 0.25 micromole, then it should be relatively safe for the cell. So it's not going to kill it because it just doesn't have this ability to meet the chlorophyll molecule within the 1 millisecond while we have this critical state. And if there would be no oxygen in the atmosphere as it was long ago when these bacteria invented photosynthesis or oxygenic photosynthesis, then the concentration be far away from the cell will be about 0. And therefore, in these Globacter, the concentration of oxygen will be point or less than 0.01 micromole, which is much less than that number. So this kind of species could basically develop oxygenic photosynthesis and not kill themselves. They didn't need any photoprotection at that time. That's basically the message here. So we could make it in a stepwise manner. So excess oxygen is less than 0.01% of atmospheric today. And indeed, oxygenic photosynthesis may have evolved first, and then oxygen photoprotection or all the protection mechanisms would have developed later during hundreds of billions of years, along with the rising concentration of oxygen in the atmosphere. So it looks more like a patching then. So nature invented something, and then you start patching things and fixing things, and something breaks again, and you patch that. Because some time ago, I was always thinking, well, nature did it the best way possible, but now I'm kind of more thinking that it's more about patching, because that's the easiest way to do something. So this is all I wanted to talk today about. This is the acknowledgement. This was done in my physics department. These are my students working on the problem. And we had a good collaboration with Bill Kleiner on the BChl complex. And of course, we have collaboration with Don Bryant's group at Penn State with the Chlorosome, the mutants which didn't have any weapons. And with that, I would like to end. These are the conclusions listed, which I talked today about, but you can read all this stuff. Thank you very much.
Now it's on. Any questions? Good.
So the 1 microsecond quenching time that you quote obviously is for typical 2 millimolar or something oxygen in solution, and the usual rate constants, like you said. But in the cytochrome BF, to get that same sort of 1 microsecond in which you have the oxygen being the mediator, then the assumption is that it's still on that same order of concentration, or else it's a locally higher concentration because of these hydrophobic interactions or something?
It's a good question. Yes. So there was a hydrophobic channel, and what we assume is that the oxygen is pretty much trapped in that channel. It has to be. So the local concentration should be higher. And of course, the concentration there would be already 10 times higher in general because it's a hydrophobic environment. If you go from water to methanol, for example, the polar, non-polar, you get 10 times more oxygen in methanol than in water. But here, we should actually have it in and sitting there. And this is exactly what X-ray shows with xenon. But it's expected to be a good model for oxygen. It's just we can't see oxygen in X-ray. But xenon was right next to the chlorophyll sitting there. So it has to be in high concentration sitting on it. There is simply no other way you can transfer a triplet state here. And currently, we can see oxygen dependence as well.
So you had the calculations for chlorophyll molecules inside a grid, but what do you think of the edge effect that will have on the energy of the triplet state? So if you, I think you consider a grid of molecules and calculate the triplet state energy as 0.9.
Excitonic energy? Yes.
Say again?
You saying excitonic calculation?
Yeah.
Yes.
So you consider it as a grid, right, for calculation?
Yes, very simple, because we don't really know all the numbers. It was more like a ballpark calculation.
So what do you think that the triplet energy will go right? I mean, will increase on the edges of the grid? Or?
It could be, but then again, if it has overlap with other triplet states or excitonic states, there would be transfer of energy to these lowest states. It's just triplet transfer, but these things are overlapping. So I would expect it goes out, and it's mostly bulk, really. There is not so much of a surface. Now, in principle, some of these, if you look at these quenching of the triplet phase of chlorophyll, you can actually see there is a passive component as well. It has a small amplitude in the beginning. That could be quenched by oxygen. But the number of those should be small because the system is just so stable, amazingly stable. For triplet excitons, I should say that if you go to molecular crystal community, there are studies of molecular excitons all over. But molecular excitons, or triplet excitons, and triplet excitons have not been mentioned in photosynthesis at all. I mean, except my paper some time ago. But this is very well-known phenomenon that you do get the triplet excitons when you have them so close. And of course, the immediate consequence is that you should have lower energy states, no other way. And everything would basically match. We are actually set now to measure and see how these triplet states go in energy as we make timers and so on, and we're collaborating with Bob on that. So we just got these 7 different chlorophylls, and we got a new machine which can measure a phosphorescence, because that's the only way you can get the energy directly. So it took us quite a bit of time to make that system, because the phosphorescence has intensity of about 10 to the minus 7 compared to fluorescence, and we have to basically gate it with mechanical means, like in a few microseconds after. We have to cut the luminescence because the scale actually goes all the way to infrared. So it was, but yeah, we are on a way to actually find out what the limits are and then do calculations with some numbers, which we can compare with experiments.
Sergei, really liked your calculations about oxygen and how the concentration went up and how the photoprotection mechanisms. What about the oxygen evolving center of photosystem 2? If you do those calculations, the diffusion of oxygen out of that 4 manganese calcium oxygen center,
yes,
and by the time it escapes, local concentration of oxygen.
We tried that. So, as a matter of fact, the critical time here would be a microsecond. So how far does it diffuse? Well, actually, not a microsecond. The critical would be the time between creation of each oxygen in that system. How far away can this oxygen escape by the time next one comes in? And that was, you can actually calculate that, that was more than a micron. So it just goes away already. There is no way that this is still here unless you bind it somehow. But I doubt that the system actually likes to bind those oxygens. I think there is everything done to get rid of that immediately. But it is a micrometer or so.
I mean, this question of the local concentration of oxygen in
yes.
cyanobacteria and chloroplasts, something that's been around that people have thought about for a long time, but it's extremely difficult to actually measure experimentally, and I don't know of any good measurement. Maybe you know of some.
I saw one measurement with NMR or something like this, but this was under, basically they had too much oxygen already produced. By the time they measured it, it was like 5 times more than atmospheric produced by just that cell inside, so it was high everywhere.
So I think that we can discuss that later on. This is the common impact on many of the computing ideas.
Because I wanted to measure it, and I read everything, I tried everything, and I realized it's just not so easy to measure these small concentrations.
No, that's right. It's extremely
probably tricky. I mean, maybe it will be possible, but I couldn't invent anything. But the diffusion theory is just so simple, and it's a classical. It really works. It's like train going at this speed. How far does it get at that time? It's about that simple. And since the oxygen production rates are measured, one can really estimate it, or not even estimate, calculate it pretty precisely. Of course, the situation will be different with the colonies of cells. So we made colonies. So then when we get something like 50 micron blob, then concentration goes down. Or you green leaves, you do calculation there, and I think it's like 50, 100 microns, and that actually becomes saturated, really saturated inside. And this is like in green mats, you can see bubbling oxygen coming out. But when I was doing that, I was more thinking about the very like billions of years ago when cells are just floating around, individual cells, and how if they could actually develop this photosynthetic function, these oxygenated photosynthesis, without killing themselves immediately.
It is well known that in some cyanobacterial mats that where the oxygen has been measured, that it can be 3 or 4 times
Yes.
saturation.
Yes. We
have very high oxygen concentration, which
we calculated that. We got a very nice agreement. Once you get to certain thickness, it should bubble out.
but then you have to start worrying about things like photorespiration with Rubisco, because the sort of simple explanation about Rubisco and oxygen competition for carbon dioxide fixation says that at 3 or 4 times saturation of oxygen, you should essentially have no photosynthesis going on at all. So there's still some puzzling questions there. Do we have any outside questions? No? Do? So I wanted to ask one more question about the B6F. If has Bill or anybody tried to mutate out the chlorophyll to destroy the chlorophyll binding site, or is that lethal? Or
I would guess it's lethal, but I don't really know. I think it will basically kill it. This is like part of the structure. This is Bill's idea. Is like a basically kind of thing which holds it together. Like the keratin B is like a toothpick holding some protein.
So more a structural role.
He assumes that this is a structural role. I have some reservations about it, because I would think that if you're building a house, and you wouldn't really put one brick of dynamite instead of something that may be safer. So it would be kind of.
There it is.
And then, but that's what it is. So I think the chlorophyll is, but maybe singlet oxygen is actually some electron scavenger from heme, or like a rescue. That's what we are trying to test now. If the singlet oxygen is that channel, it actually is closed also to the hemes, the terminal hemes, and there is a possibility that if you don't have any phenols in the pool, this thing gets stuck, and you basically have to rescue it. If you take it away, then you can start running the system again. So, but I don't know. We'll try that.
So I couldn't tell from the structure, but is that chlorophyll fairly planar? The reason for asking is because if during the course of the reaction, the heme's transferring electrons, you can get some distortion of the protein, that the chlorophyll becomes at all a little non-planar, from all this stuff that we've done over the years. It could self-quench itself, so the lifetime could easily be reduced from 5 nanoseconds down to a couple of 100 picoseconds just on its own if you distort it a little bit.
That I didn't really consider.
That's very interesting, yeah.
With regard to some of the mechanisms by which, the photoprotection occur with the xanthophyll cycle, whether or not you